Good morning. Today I'd like to share with you the work that we're developing towards seed bank in-depth characterization. This work is being done in collaboration between the most important Arachis seed banks in the world, the ones with the most expertise. It builds on the long experience of the institutes in Brazil and Argentina, the curators and researchers who dedicate their careers to understand and utilize the Arachis treasure trove, also here in the United States. This is combined with the technology that was developed as a product of the sequencing of the peanut genomes. Peanut belongs to the genus Arachis that contains 92 described species and several others still undescribed. It is endemic to South America and adapted to a variety of environments. The wild relatives of peanuts show morphological diversity, perennial life cycle, leaves with 3 leaflets, tuberous roots, hydrophyly, and many other characteristics that are very different from the peanut we all know. From the late 50s until recently, many trips have been made in South America, mostly Argentina, Brazil, Bolivia, Paraguay, to collect wild Arachis species, generating a great body of knowledge, an invaluable asset to the research and breeding communities. 64 species were then described, and these collections and this work resulted in the monograph. Originally, the monograph was written in Spanish by Gregory and Krapovicus, and it was later translated by David Williams and Charles Simpson into English, making more accessible to the wider community. The monograph has amazing detailed taxonomical work with anatomy, cytology, adaptation of plant form, perennial annual habits, crossability between species. It has detailed descriptions of seeds, habitats. And with all this information, the 64 species were grouped into 9 taxonomical sessions according to all these characteristics and relationships. The most known section is the Arachis, where peanut Arachis hypogea belongs to, but also has Arachis anosperma and Villosolicarpa that were used by Amerindians as a food source. We also know that there is the forage peanut from different sections, Glabrata and Calorisi. And also more recently with research in the Arachis section that is compatible with peanut, several species are being used to transfer alleles for resistance, conferring crop protection. The main wild Arachis seed banks are in South America, Argentina and Brazil, in the United States and in India. The ones in green are supported and maintained by the government. The one in blue, APRES, is an international institute. And the United States are 2 universities that hold a lot of accessions too. The USDA PGRCU holds 65 out of all Arachis species. Its primary goal is to preserve this valuable germplasm for our researchers worldwide. It's amazingly well curated, it distributes worldwide, and it provides the necessary genetic variability for different programs. So we all see the need to preserve, to understand, to characterize these Arachis collections. And we reached out to all these germplasm banks, and we got very positive responses from the curators and researchers, understanding, wanting to know more about their collections. We are collaborating with all these programs. ICRISAT hasn't been able to send material yet, but that's a work in progress. So this work aims to characterize, classify, and confirm the identification of wild species, identify new species and confirm some assignments, check for label mutations that, it's a natural thing to happen. We look into genetic drift. So there's lots of new things that we can look and understand. We want to ultimately be able to create a species barcode. So when we have a new species or something we don't know what it is, we'll be able to, by genotyping it, be able to assign to a known taxon. This clearly is not a new idea. Various authors, including ourselves, have published research using anatomical, biochemical, molecular markers to detangle the genus structure. But now we have the technology that really allows us to assay an unprecedented number of markers genome-wide. And this barcode will be an extra tool to help with the identification. And it's going to be especially useful in places away from the main traditional expertise. So at the moment we have 6 institutes working together. We have been able to get DNA or plant or dried leaf from different places. We have some accessions that are in common between different institutes. So far we have 91 species. Some of them are not identified yet. 368 accessions, and in total 725 samples. So after genotyping these materials, so we extract DNA, we receive materials, extract DNA, genotype, analyze, compare. So it's a long process, and it's been happening for the last 2 years. Our first results, so far, we're going to concentrate on the Arachis section, which is the most important one for the utilization. And it's the one we are concentrating first. So from this 562 samples, we've got some groupings, and the groupings confirm the different genomes, subgenomes. So we have the A genome, B genome, F, D, K, etc. And the clusters are basically, with a couple of exceptions, they confirm the taxonomical separation. And this is a good calibration for the method. It shows that the SNPs are informative and are consistent. So again, working on the phylogenetic tree, on the Arachis section, so we see the groupings are as expected for the most part. This is removing the redundancies. So each point of these is a different accession. So we start looking at the peanut progenitors, Arachis ipaensis and duranensis. We have Arachis magna, which is a cone species from ipaensis. Arachis cardenasii, again from the A genome. Arachis cardenasii is the famous donor of multiple resistances in Nematode, Tifgard, Bailey, and all the cultivars around the world. We have Arachis batizokoi that we've been working with from the K genome. And we have an interesting case of Arachis colmani. Arachis colmani is native to Brazil, and it's been collected in different geographical areas. And when we look in the tree, it clusters in very different clusters. So they're different groups. And these different groups, they are correlated with their geographical origin. And this is being consistent with previous work. And it seems that it's so well separated and quite far in the phylogenetic tree. This may be a case where a species concept need to be reevaluated. And finally, we have the case of Arachis stenosperma. This is a favorite of ours. It's a species that we've been working with for a number of years now. It has alleles that confirm multiple resistances. We are releasing germplasm this year with stenosperma in its pedigree, and it will be accessible to breeding programs and research. So if we zoom in the Arachis stenosperma clade, we see that this is again endemic to Brazil, to the region of Mato Grosso and Goiás, which is right in the middle, and also in the coastal regions. When you look at the tree, it is clustered into 3 adjacent groups. They are the same group, but they are clearly distinct. When we look at the geographical origin, again, we see that it really is consistent with their origin. But let's look very closely. If you look at this group here, it's very basal, which means it's the oldest group. This basal group is more in the west and north. If you look at the intermediate group, it's a little bit more south and east. And if you look at the most derived group, it's all the way into the coast, very far away, about 1,000 miles away. So zooming out, what's really interesting is that this genotyping confirms the Arachis stenosperma distribution that was done by Amerindians. This is a theory that Amerindians used the Arachis stenosperma as a food source, and they migrated from the center of Brazil all the way to the coast, and taking seeds with them. So when they arrived at the coast and they settled, they spread seeds and created populations there. This story was unraveled by Dr. Vals, and now we've been able to confirm it. So it's been a very interesting journey. We are genotyping results that agree with identification. We have new collections with new species assignments, and we see difference between annual and perennial species. And it's created a lot of good answers and very interesting new questions. So it's a work in progress, and these are the main actors of this group, of this work in alphabetical order. But I'd like to give special thanks to Carolina Chavarro, who was the one who really is doing all the heavy lifting of this work. And I'd also like to thank our funders. Thank you very much.